Okay, I'm still waiting.
It's saying preparing. It's saying preparing.
Yeah, yeah. Yes, we have connection. And we are live now.
Yeah, yeah. We are live on YouTube. We are live on YouTube now.
It's 11 o'clock on Sunday morning and time for yet another Chai and Why. Today is the 5th Sunday of the month, a special month which has 5 Sundays. And our 5th Sunday Chai and Why has always been something which has a hands-on component that people could really participate in and do stuff with us. Unfortunately, because of the pandemic, we have to do these sessions online. We aren't meeting physically as we always used to. But this session is really, really special. This is not just a hands-on session, but a distributed hands-on session with participants from across the country. And for this, we've invited the Cube project people. Now, what is Cube? Well, it's not a sphere or a rectangle. Don't think geometry. Cube is something different. The C is for collaboration, which is something very, very important in these times. And we will have Professor Nagarjun explain what Cube is in a bit. But before I hand it over to him, just remember that you are all watching this either on Zoom or YouTube or Facebook. The only way we would like to make this as interactive as possible, we would love it if you asked us questions, put in your comments. So please, please use the chat box if you're on YouTube and Facebook. On Zoom, you can put it, if you have questions, you can put it in the Q&A box. You can also use the chat feature. We will try our best to answer your questions. We will, if it's really, really important, we will interrupt and ask the speaker. Otherwise, we'll wait for them to finish their experiments or whatever they're showing us, and then we will take your questions. So we would love your questions. Keep asking questions. So with that, without wasting any further time, let me go ahead and introduce Professor G. Nagarjun, GN. GN was at the Homi Bhabha Center for Science Education, which is part of TIFR, till just a few months back. And he is the one who's responsible for along with others, for setting up the Cube project and many other things that involve the public at large in experiencing science in a very easy to do in a hands-on manner. So over to you, GN. Enjoy the show.
Yeah. Thank you, Arnab. It's been great to be back on Chai and Why, and particularly because many of the Cube students are doing wonderful work during this lockdown or pandemic. So I must say that, thank you so much for giving them this opportunity. So I will take a little bit of time to explain the various questions about not only about how to make a home lab. That part, of course, the students are going to make. I will be mostly talking about why make a home lab, and also what do we do and what is a home lab, and also what is a Cube about. A little bit, so that you all get some kind of an idea about what it is. It's a collaboratively understanding biology education. That's just an acronym. So it's not a geometrical object. It's a social network of scientists or budding scientists, you may want to say. And of course, we started as a collaboration in 2011. With Sophia College, where the primary energy of the Cube began with Professor Arunan. He is the seed, the root, and the stem and the branches of this project. And we sort of started this collaboration, and then HBCSE TIFR is hosting this project since then. And meanwhile, of course, we have collaboration from other organizations, particularly Kishore Bharati, and of course, not last but not the least, all the CUBE participants. I mean, on the whole, we have already more than 2,000 to 3,000 participants from across the country during these very close to a decade.
GN, maybe you should just say what is Kishore Bharati. The age group watching this probably doesn't even know about how it got founded and where it is from.
Yeah, well, so let me say that. I mean, since you mentioned, you see, Kishore Bharati is founded by Anil Sadgopal, who was a scientist in Obed Siddiqui's lab at TIFR. That was in 1969-70. And it was at that time that Anil started chatting with people around, and with some support after 2-3 years of his being a scientist at TIFR molecular biology lab, he left to start Kishore Bharati to the villages in Madhya Pradesh, particularly Hoshangabad. So some people in the audience may already know that HSTP project, as it is sometimes called, the Hoshangabad Science Experiment. And so Kishore Bharati has adopted CUBE as something that is continuing that tradition of activity-based education. And so, yeah, thanks Arnab for giving me this opportunity to say a few words about Kishore Bharati as well. So where do we do? I mean, as Arnab already mentioned, we are doing across the country. And once in a while, we also get participants from other continents as well. And of course, so this is kind of all the collaborators, and some of them are already started having these home labs. And this is a spread of our CUBE participants, and it keeps changing from time to time, but on the whole, that is where we have reached so far. And we certainly want to increase the density with all the collaboration, all the participants who are listening to this, please do join us. And then the main question, of course, is that, you know, what do we do every day? And I'm underlining the word every day because we do it every day. So we work every day in home labs, and we chat every day online in the ChatShaala. I mean, I mean, you can see the pun of the word. It is not Pathshala, it is ChatShaala, because we think that conversation is the key for engagement. And then we publish every day. That's what, I mean, sometimes we can call it as we share every day. And then we seek feedback from the other budding scientists every day on the social media. Of course, these are some examples of the social media that we use. So we have metastudio.org, which is our platform, which is hosted at HBCSE for supporting this large social group of scientists from across the country, which has students, teachers, and sometimes even enthusiastic citizens and curious citizens who just join and start working with us on these investigations. And we also do have a makerspace at this place. And so the CUBE webinar room of HBCSE, also hosted at HBCSE, becomes live every day at 5:30. I must mention, every day at 5:30. And starting last April, we have completed about 527. Today's will be 527th CUBE webinar that's going to happen, and it's for 4 hours a day. And I must also thank here for this success, Kiran, who is in the room here as one of the volunteers, has not taken a single day off for all these 527 days. So she starts the room and then continues for 4 hours on a regular basis, wherever she is, whether she is at HBCSE, where she has gone home, working from home. And it is not without her help that we would have managed all this. Of course, we have WhatsApp groups and Telegram groups, and of course, email as well. So we use all methods of communication because we think that communication is the essence of science in a major way. Okay, so now the question, I think the most important thing, I will certainly want to ask this question: why home labs? I mean, because a lot of people think that home lab is a necessity because during the time of lockdown, all of us are working from home, And therefore, you may be thinking that home labs are necessary. But then, we actually think that, home labs are necessary from the beginning. So that's how the Cube, when we started in 2011, we encouraged all the students to actually start home labs right in the beginning. And that's actually one of the reasons why we say that it's a pedagogical necessity. And also a constitutional necessity, because India is a unique country, has a constitution in which it is written that every citizen must develop scientific temper. But the question is, how do we reach every citizen? And that's a big and $1 million question. So Cube is an experiment. So we started with the model. And so what is that model? I'll just briefly spend a couple of minutes tell you about that. So we know that India is world-class when it comes to cricket. So our hypothesis is that we became world-class in cricket because we play cricket in the streets, in every street corner, in every mohalla. So taking this particular clue from the cricket as something that is played on every street, that's also the reason why we have no depth of excellence in that game. And we have, that is the important aspect of that. Now, can we actually do this kind of thing? Can we take Cube, or can we take STEM activities, science, technology, engineering, and mathematics activities to every street corner? Can we have a club? Can we have a STEM club everywhere? So that's the idea behind that, and that's the small. So here is an ideal game, like an ecosystem. It's a kind of a pyramid structure that maybe you must have seen in your lectures So at the base, you have the producers, the people, who contribute the energy. And some of the people not only play the game, they also are actually the spectators. They understand the rules of the game. And some of them actually play the game so that the whole country can watch them. And then out of those players, some of them actually become the referees and coaches. And so this is like of course, the size of the rectangle basically tells you the number of participants. Of course, you always have very few people who would be doing that. But so what is the most important thing that we need to understand about it? So when it comes to science or STEM in India, so this is the picture. I'm just showing the contrast here. The picture here is that, somehow in India, there are very few or absolutely no spectators when it comes to this. That's one possible reason, is that the way how our higher education institutes are structured, we don't do it in front of everybody. And that's one of the important things that we look at here. And so how important spectators are is one of our hypothetical claim. So Cube uses that as an important cue. So, and therefore, can we play STEM games like cricket? Of course, I'm just using cricket as a metaphor here. People in the Northeast would use football. Maybe also in Goa and Kerala, for example, there are a lot of fans for football as well. So it depends on what game you play in the street. And that is what was really an important aspect of that. So this is the lesson from the cricket that we want to carry on to STEM education. So how do we create spectators for STEM games? So it's here now. I will over to Professor Arunan. Please tell us on why spectators are important. Over to Arunan.
Hello. Thanks, Jian. I think I will remind the audience of 2 points Jian has emphasized. One is that there is an educational necessity and a constitutional necessity. That's first point. Second is that he proposed a model when the spectators are remote, it almost collapses or not as expected to, as much that we expected it to function. So how do we, so can we illustrate that model in some way? So I would like to show this picture, a few pictures, and here is one picture, and we will play a small game for a little while. So I would like people to say about who is this? Can we recognize this face? Can we do a poll to say that yes or no? So some time is given.
Okay, so here's a poll. All you have to tell us is, do you recognize this person? Yes or no. If you're on YouTube or Facebook, just put in comments, yes or no. Do you recognize this person? We'll give it 10 more seconds. Oh. This is not looking good. 17 out of 20 people. Let's wait till we get to 25 maybe. I see a lot of comments on the chat as well. So only 3 people have said that they have in the poll. Okay, I'm going to end this poll now. So only 3 people said that they recognized the person, and most people, the rest of them did not. And let's see, in the chat also, I think most people have just said no. So yeah, so I would say that okay, there's another picture coming up.
Yes.
Another picture coming up.
Yes.
Let's do
yes or no. Do we recognize?
Let's do another yes or no. Let's do another yes or no. How about this person? Let's see if this is any better. Oh, now we are getting yeses. Let me move this poll box away. How do I move this poll? Oh, come on. The poll box covers the face. Isn't that terrible? He seems familiar. Yes, he should be familiar. He should definitely be familiar. Somebody says he seems familiar. And okay, where did the poll go? There it is. Oh, in this case, now the yeses are about, I would say, 68% to 70 to 30, yes to no. Okay, so certainly in the first one, we did very badly. Hardly anybody said that they recognize the person. At least this one, people have said 70% of the people say yes, 30% say no. Let me quickly check how YouTube is doing. Okay, we are.
How about this? I was not.
Okay, let's see. Is there anybody who doesn't recognize this? Let's put it that way. Let's see. If you do not recognize this person, say no. Otherwise, don't even bother.
Don't even bother.
So anyone who does not recognize this person,
don't even bother.
We're getting flooded with yeses. We're getting just flooded with yeses on the chat. Okay, you recognize clearly. Oh, the guy who lost the last test. That's a nice way of putting it. So, yes. Arunan,
in spite of
Arunan, we have a problem, right? I mean, everybody seems to recognize a cricketer, but not a scientist.
That's not a problem. No, that's the actual thing.
So that's where we are. You see what we've just proven to you?
Yes. So
what did we just show?
That's where we are. That's where we are. So one of the reasons probably could be this: every nook and corner, every mohalla you see people playing, if not as Virat Kohli, but as themselves, and probably imitating, emulating. So every corner, you may be seeing it. Okay, so not only they are playing, but there is a large number of spectators many times. So where are the spectators for science is the question that we have been discussing about. So you look at this out, and there is a hands going up, and exactly like in a match. So the rules of the game, in every nook and corner, they are trying to understand that. Yes. Facilities. We complain about facilities in colleges, schools. Facilities. No complaints. Improvisations of all sorts. But still they play. Yes. No holds barred. Every nook and corner. Locality clubs. and look at the smart feel that the person emanates. The feel of a test cricket. And again, nothing can stop. In 2 matches, 3 matches, 2 games, 3 games, everybody can find some field space. Yes. Yes, look at the improvisation. Now, anything can substitute the necessary things. You have things here, 2 examples here, and look at the face, the happiness. So perhaps more fun by substituting things. So no wonder that Virat Kohli hits almost 100% or cricket in spite of the debacle. And where is this gentleman? Venkataraman Ramakrishnan, probably the last Indian to get a Nobel Prize some 10 years ago, 2009. But very, very few people in the general, of course, we have 68%, I think we had, but very few people in our daily experience, very few people recognize him or his work. He started as a physicist, got Nobel Prize in chemistry for the work that is often cited by biologists and by biologists. So, populating with STEM, science, technology, engineering, mathematics spectators. This is what Nagarjuna was referring to in his model. How do we create the spectators? Something like we have been able to create in the case of cricket or football in some places. So Cube's gully cricket model. So we have extremely wonderful people across the country, as Nagarjuna has shown, out of which a few people will be presenting this time, and over to Sayada for the Cube Students Initiative in Home Labs. Over to Sayada.
So, Arunand, there's a question. Who was the first photograph?
Oh, sorry. I'm sorry. That's Venkataraman Ramakrishnan when he was young, brought up in Baroda and went to US to do his PhD. Sorry.
So that was just a young
Venki. Yeah, young Venki.
Okay, with this, it's going to be over to our host for today. Our host for today is Sayada. She's a student, a BSc student, and she will be actually hosting us and taking us through the adventures of all the Cubists, or the Cube participants as they call themselves. So over to you, all yours.
Thank you, sir. Thank you. Thank you, Arunand sir and Arnab sir. So hi everyone, I am Sayada, an undergraduate student pursuing biotechnology from Elphinstone College, Mumbai. So like Arunand sir said before, that Cube as a gully cricket model, we are trying to popularize science, trying to create spectators through frugality of the lab. So let's see how Cubists are adapting to this gully cricket model. So I'm sure that we all have seen this fruit flies on fruits, fruit baskets, but we don't seem to take a note of it. So why Cubists are interested in fruit flies? So to study fruit flies, Cubist school students started attracting flies using bananas, tomatoes. Few Cubists were getting fruit flies during the day, while others were not getting as many fruit flies as other Cubists during the night. So around 5 years ago, Cubists started taking interest in this to find out when is the time when fruit fly is maximum attracted to the fruit. So they started observing in plastic bottles, fruit flies in plastic bottles. There is a fruit kept in the plastic bottle to see at what time maximum fruit flies are attracted to fruit. So by the results that we found, fruit flies were active during the day and not active during the night. So in 2017, when the Nobel Prize for Physiology or Medicine was announced for the discovery of circadian rhythm, the molecular mechanism of circadian rhythm, there were messages floating around in Cube groups that we have got Nobel Prize, because as Arunand sir said about the spectators in science, we could recognize the Nobel Prize, as we had some work. We could relate to Nobel Prize. We had some work in flies, that we were observing flies during the day and during the night. Perhaps we identified the pattern that is repeating. And when the Nobel Prize was awarded, we felt that our work is also being fetched as a prize, as a Nobel Prize. So this work done 5 years ago was extended to home. Like you see, there was a bottle kept in order to observe fruit fly. So the home lab was established before pandemic. Binita, a cubist from Ranchi, was the first one to establish home lab, popularized as kitchen lab. So Binita, can you tell us about how you developed your home lab 4 years ago?
Yeah. Hi, I'm Binita. So in June 2017, 2 months after I returned back from Cube Lab, from Homi Bhabha Center for Science Education, I started the first kitchen lab. Undergrad research is not yet popular in Jharkhand. So except Dr. Ganesh Baski and Dr. Shalini Lal, other people were really skeptical about undergrad research. So they asked me to wait till my PhD starts. And it was a bit difficult, and I wanted to check the hypothesis that we discussed in the lab. So I convinced my parents and my whole family, because I'm a part of a huge joint family. So soon I realized that the space was not needed. The space was actually needed for other people to discuss and perform science. We did several experiments in the space that you're seeing in your screen right now. And we have used pressure cooker as autoclave, and for cleaning. Milk bottles are used for fruit fly cultures. We had several model organisms, which includes fruit flies, earthworm, moina, and zebrafish, which we collected from different places over the time. And here are a few more pictures I would like to show you. So here's me catching zebra fishes from Jaldhaka rivulet. So Jaldhaka rivulet is in West Bengal, and these fishes actually traveled some 700 kilometers with me to reach Ranchi. So, and then here's Richel, who is collecting zooplankton from Jharkhand fisheries. Then here's the last picture from a cold December morning when model organisms are basking in sun to maintain temperature. 10 months after the kitchen lab was established, we actually got a space from college. So if you ever think that there's a problem because of which you're not able to perform science, I'm sure the problem is in the head. Also, I strongly believe that places and surroundings should not decide that how fit we are for doing what we really want to do. So after this, I got a chance to go and work in CCMB and do my master's thesis there with Dr. Rakesh Mishra. Currently, I'm taking a break and applying for a PhD, and I look forward to pursue science. Thank you.
So as you saw, the home lab was existing way before pandemic. Cube Lab in Homi Bhabha Center for Science Education, here you can see displayed on the screen. To start with, it was a frugal lab. See, you can see instead of using flask or beaker, we are using plastic bottles. In the back, you can see there is a pressure cooker cap. So instead of autoclave, we used to use pressure cooker for sterilizing things. So it was comparatively easier for you Cubists to continue their work when the lockdown was announced. And here there would be some home lab photographs from our collaborator. Yes, so in the left you can see this is our home lab by Ashwiti, one of our collaborator in Thrissur, Kerala. And on the right you can see home lab from Siddhi, our collaborator from again Thrissur, Kerala. So you can see the space that we get, we've converted into a lab. Then there is a clip of Raymond's lab that was established by students Manmasi, Rachel Turkey, Manjwil Jojo in Kokar, Ranchi, Jharkhand. Man, can you tell us about your lab, Man?
Thank you, Sayada. Hi, I am Manmasi Bek, and I've done my bachelor's from Dr. Shyama Prasad Mukherjee University, Ranchi, Jharkhand. So here in the video, I'm actually demonstrating a simple scientific daily exercise that Cubists and I practice daily at our respective home labs. So here in the video, you can see I'm maintaining fruit flies in Amul Cool bottles at home, which includes observation, recording my observations, and also share my observations to other cubists for their collaboration. So after observing, you can see I note them down in a logbook so as to maintain a record of my work and also to not forget my observation. Since just noting them down is not good enough, so I make sure that I've got the evidences of my observations to show them to the world. And you can see that my phone does not have an inbuilt macro lens, so I actually use a homemade macro lens, which can be prepared by spending less than 100 rupees actually. So in my case, this lens is prepared using a lens from a toy laser light, a hair clip, a sellotape, and my phone cover, of course. So we can see a close-up larval video of fruit fly after fixing this on my phone, so the larval video, the size of the larva is between 1 mm to 3 mm in size. So you can see the larval video, and after this you can see a pupa photo, which is of 2 mm to 3 mm in size. So after this video you can see that as well. So this way actually I practice to record my observations and share this to my other fellow cubists. So thank you, Sayada, for inviting me here.
So like Man said, we are observing fruit fly pupa, larvae with homemade things like using a lens from a toy, like he said. So like Man, there are other collaborators as well who are maintaining fruit fly and doing similar things through collaboration. So I'll continue with during lockdown, one of our collaborator Ashwati from Thrissur, Kerala was meticulously looking into the fruit fly activity again, because when we were observing fruit flies, we used to get that okay, after evening the number of flies is reducing. So we used to expect in the night will happen the same. So she meticulously looked into it by being awake at night to find out whether fruit flies are really not active during night. So these are some observations during the day. Ashwati, a third year BSc botany student from Thrissur, Kerala. Can you tell us about your findings, Ashwati?
Yes, Sayada. So actually, I got more number of flies during the daytime and less number of flies during the night. But while observing fruit flies on fruit, also found several other organisms like mosquito, house flies, worms, etc., along with fruit flies. So to be further study about fruit flies, we need to culture them, specifically isolating one single pregnant female so that all progenies coming from the same mother will be same species. Now I am going to show my home lab. I hope everyone can able to see this culture bottle.
Yes. Yeah.
Yes, Sayada. So these are the culture bottles that I am maintaining in my home lab, Guttukadavu, Thrissur, Kerala. So, these are the culture well started from during the lockdown period started, and this all culture bottle started with one pregnant female fly. And every month I'm transferring these flies into new media bottles in order to maintain the culture. Like me, several other collaborators from across the country are maintaining this culture at their home lab. Over to Sayada.
So yeah, thank you, Ashwati, for giving us the tour of your home lab. So like Ashwati said, we are maintaining fruit flies at home, but actually before lockdown, we used to culture flies in the standard corn meal agar media. What is shown on the screen, this is standard corn meal agar media, and we are putting it in a Amul Cool bottle, again frugal lab. So this is how we used to culture flies in lab. But what do we do at home? How do we culture flies at home? Can you tell us, Yash? So Yash is a postgraduate student from Prabhadevi, Mumbai. Yash, can you tell us how do we culture flies at home?
Hi, Yash, are you there?
Yeah, hello everyone. Myself Yash, I am an MSc postgraduate student from Prabhadevi, Mumbai. So as you can see on the screen, there is a map which is actually map of Cubists from all over the country, like Bihar, Jharkhand, Kerala, Assam, who regularly participated in everyday ChatShaala discussions, as you saw, and came up with a solution for a homemade medium by substituting the ingredients from standard corn meal agar medium, right? So I hope you'll see a screen now of the regular ChatShaala discussions that we do. And this is a discussion where we were discussing about how we can prepare a homemade medium by substituting the ingredients of a standard corn meal agar medium, as Sayada said, where we culture fruit flies. So as we know, tomato is actually a fruit where fruit flies are naturally attracted to. And rava, suji or semolina is actually a substitute for agar in the medium, which is used as a solidifying agent, right? Sugar is actually the table sugar that we have in our house is actually a substitute for sucrose that is present in the medium. And while actually we actually prepared the medium and we observed a lot of fungal growth in the medium. So while discussing, that's when we came up with an idea from pickles. So as we all know, pickles we eat, but we don't, when you will be knowing that pickles have vinegar, which is acting as a preservative. So actually, why not use vinegar as a substitute as an antifungal agent in our medium? So that's what we did. And also you could see on the last 2 points, which is there in the standard corn meal agar medium, propionic acid and orthophosphoric acid. These are substituted by vinegar, which is actually acetic acid, and it's quite similar to propionic acid. So that's how we prepare homemade medium, which we popularly say it as tomato rava sugar vinegar medium or the TRSV medium. So right now, actually, I will give you a tour to my kitchen media preparation setup. So let me just take you all. So this is a long shot of my kitchen right now. I will show you all how I prepare generally the medium, the tomato rava sugar medium. Here, I hope it's visible, my ingredients for tomato rava sugar medium. As you can see, I've taken an egg-sized tomato, normal egg-sized tomato. What I will be doing is I will be chopping this tomato into small pieces. And actually, I've kept the things already ready so that I save time. And other ingredients, as I was mentioning, here you could see rava or semolina or suji, which is acting as an agar solidifying agent. Here is the table sugar. And what I will be doing is in any medium as we require water, right? But since we are in our home labs where it's just a kitchen, we don't have sophisticated instruments like burettes, pipettes to measure water, right? And what we've done is I've used sanitizer bottle. So why sanitizer bottle? Because it is popular, and what the thing is that sanitizer bottle has ready-made 100 ml, as a unit. So I'm using this a frugal, measuring unit for measuring water. This is my pan. What I'll do is I need 150 ml of the water. So I will take one full of this and half of this to add water first in the medium. I will turn the flame on and I will add the ingredients. What I'm taking is a tablespoon, 2 tablespoons of rava, 1 tablespoon of sugar. That's the ingredient that we came up with. And later on, I will be adding the tomato. So, and then after once the medium is cooked for 10 minutes, we'll actually add vinegar. So let's actually see a video so that it will be much clearer. And we have a video of our collaborator, Isha. Will just be visible from Isha from Worli, Mumbai. And she is demonstrating how we can make TRSV medium, right? So as you could see on the screen, there is the ingredients of the medium. So we require generally 150 ml of water, and we add 2 tablespoons of rava to the pan, 1 tablespoon of sugar, mix the medium continuously and cook the medium like we take notes from our mother, who helped us in actually cooking the medium also. Then we add tomato. Once cooking is done, we add vinegar and stir the medium, right? Let it cool, add vinegar, then transfer the medium using just a spoon, or if you have a funnel, you can do it in our bottles. You can use plastic bottles as well. Here it's a glass bottle, and definitely cotton plug it so that nothing can come inside. So that's how you could see we prepare homemade medium. And here is actually a picture of another collaborator, Raj Patil from New Delhi, and he's actually prepared tomato rava sugar vinegar medium for culturing fruit flies. So it's that easy to actually prepare and culture fruit flies in our home lab. Yeah.
So
Thank you,
like
everyone.
Yash demonstrated and showed the video that how we culture flies at home using this tomato rava sugar vinegar medium. We are very sure that the flies are from a single mother, like Ashwati added. They are of the same species, but are these Drosophila melanogaster? How do we identify a fly? Are these the well-known fruit fly that got 6 Nobel Prizes? So how do we go about identifying a fly? How do we find out whether these are Drosophila melanogaster or not? So Isha, can you tell us? Isha is a third-year BSc Biotech student from Mumbai. Isha, can you help us identify our fruit flies, Isha?
Yes, so hello everyone. As Sayada said, we will first have to identify whether a fruit fly belongs to the same family as Drosophila melanogaster. That is the standard fruit fly. For which,
Isha, do you want to turn your video on or your bandwidth is low?
Yes.
Go ahead. If it's a problem, it's fine.
Yes, so for which we have to look at the finer details of the wing of the fly, especially the two bricks, along with other features, as you can see on the whiteboard of the ChatShaala discussion. Usually in college labs, we use a mobile microscope, but in home labs, microscope is not available. So through collaborative discussion in ChatShaala, we developed our phone camera as an alternative to the microscope. Now, let's understand microscopy by using a phone camera, by taking a look on how do we convert a phone camera into microscope at home. So as we can see, here's the setup of the phone microscopy. Starting from the bottom, first we place a torch, then above the torch we place a white sheet paper, and above that we place a glass sheet. Now we take an object that is the immobilized fly. So how do we immobilize a fly? Just keep the fly in the refrigerator for an hour, and after then we can use it for identification. Then we will take a mobile camera and we can adjust the focus accordingly. As we can see in the video, we can zoom in and zoom out to get the clear picture of the fly. Here's the picture of the fly. We can see the finer details if we zoom in. And
So,
here's
thank you, Isha. So this we'll critically look at this photograph, look at the two breaks that Isha talked about, and identify whether this fly that we have that we are culturing is belongs to Drosophilidae family or not. So, along with identifying fruit flies through phone microscopy, we have been maintaining fruit fly cultures like Yash and Ashwati and Isha said in tomato rava sugar vinegar medium. And in one of our culture maintained by our collaborator Ashwati from Thrissur, Kerala, the fruit flies were declining in number. Here you can see very less flies can be seen in this bottle. So to find out what was the goof up, what was the mistake because of which fruit flies were declining in number, we had some Alexander Fleming moment. So when we took a closer look at these bottles, there were small worms that were growing in the medium along with fruit fly. So as you can see, these worms, actually, if you see, these worms are not as, it is very small. It is not as big as the larvae or the pupae that is shown by Man in his cube labs, in his cube home lab. So when we were observing this worm, we also saw that the movement of this worm is very similar to that of the nematode, which other cubists are working on and finding it difficult to culture. So by this, by celebration goof up, by understanding, by popularizing our mistakes, we took a closer look at the culture and we found a medium for nematodes. So like fruit fly, C. elegans, a star nematode, has been awarded 3 Nobel Prizes. But it has been reported that C. elegans is not found in India. As you can see on the screen, C. brigaceae is reported in India, but C. elegans is not yet reported in India. So Cubists say that we have not looked enough. That's why we have not found. So Cubists across country are continuing the hunt to find C. elegans in India, in Indian native soil. So in college lab, when we were doing this, we used to use E. coli culture, as nematode used to feed on bacteria on an agar plate. So we are keeping soil at the periphery, and in the center we'll add the E. coli, so that nematodes will attract towards the E. coli, as it is the food for the nematode, and that's how we can isolate the nematodes. But in home lab, again, we don't have agar. How do we culture it? Before this TRSV medium before this Alexander Fleming moment, we noticed what we used to do is Batul Mam will tell us how we used to culture and isolate nematode in home. So Batul Mam from Kolkata, West Bengal, can you tell us how do we isolate nematodes at home? Batul Mam, over to you.
Hello, I'm Batul from Kolkata, and I will show you my home lab. As you can see in my home lab, it's a very simple home lab having raw potato slices, a magnifying glass, a toothpick to pick the worms, soil, milk, and water. What we can do is to culture nematodes. If we do not have agar at home, we can use raw potato slices. On the raw potato slices, I have put milk at the soil at the periphery, and the center I'm adding a few drops of milk. The bacteria from the milk will use the potato as a nutrient and will multiply. And as they multiply, the nematodes, whose food is bacteria and yeast, will come out from the soil. I have put another slice also, that will be the control slice, on which I will just put the milk, and we won't put the soil on that. After 2 days, you will find nematodes coming wherever there is microbial growth on the potato slice. I'll show you a slice. This slice is around 12 to 14 days old. And in this slice, I can see nematodes growing. They are multiplying over there. I use a magnifying glass. I use a magnifying glass very close to my phone camera because nematodes are very small. They are 1 or 2 millimeters in size. So to be able to see them, I use the magnifying glass close to my phone camera and focus it and zoom it with my mobile camera to be able to see the nematodes growing. I will show you a video in which you can see the nematodes growing.
So like Batul Ma'am said, she's using a lens that is a lens with the camera, with the phone camera, to observe the nematodes. But with the Zoom meeting, actually, we cannot zoom in to zoom out, or that is why we are showing a video of how she actually observes nematode using in her home lab using phone.
So in this video, we can see the nematodes, which are 1 or 2 millimeters in size, and they are transparent. They are growing in a very large number on the potato slice. You get a very good culture on the potato slice. They can then be transferred to a fresh potato slice. If your potato gets, as you saw, a big hole has developed in the center of the potato slice, a crater. As the nematodes are multiplying, you can transfer it on another slice after it grows. Here we are observing it. And this is me working in the home lab.
Yeah, observing nematodes. So like Batul Ma'am said, that on the potato slice, milk is added and the bacteria in the milk will grow, considering potato as a medium for them to grow, and nematodes would be attracted to it. So why can't we study conventional microbiology in home labs using this frugality? For example, with the help of toddy or fermented coconut water, we can study yeast. Aditya, a second year MSc student from Panvel, Maharashtra. Can you tell us how do we study conventional microbiology in home lab? Aditya.
Yes, Sayada. Hope I'm audible.
Yes, Aditya.
Yes, do you want to turn your video on?
No, sir. I will be just saying some few lines and then I will turn on my video, sir.
Yeah,
So the toddy.
You can just show us your
fermented
face.
coconut-
People can see you. That's all.
Okay, sir.
You can flip your video camera to show the lab.
Yes, sir. Okay, am I visible?
We can see some plants. We can't see you right now. You'll have to change your move to the front. Yeah, fantastic. We can see you. Great. Makes a big difference to see the speaker. Thank you.
Yes, sir.
Go ahead.
Okay, sir. Okay, sir. So fermented coconut water is what we can say a substitute to toddy. What we can say, the toddy is traditionally made from the sap, collecting the sap from the unopened coconut flower. The cut is given. We can see in the picture the people are climbing over the tree. They are giving a cut towards the inflorescence, and a small earthen pot is hung there to collect the dropping sap, and this sap or juice is then fermented, which gives rise to the alcoholic drink toddy. So in home lab, we were discussing, can we use fermented coconut water as to study the fermentation process and some microbes related to it. So in home lab, what we do is like we simply take the coconut, break open it, collect the water, coconut water, and fill the coconut water in the glass bottle or plastic bottle, whichever is available, and keep it for some time or some hours to ferment, and then we can spread it on the potato slice and observe the microbial growth. So I would try to demonstrate my home lab also and the process also. I'll just flip to my camera. Okay, is my home lab visible now? I hope.
Yes.
Yes. So these are the phyllanthus plants which I am also maintaining it. So as we can see, it's a common weed. I would rather say a significant weed, which we will be seeing in upcoming time within a few time. And I'm culturing them. I'm maintaining them in simple containers. Like this one is a plastic box which was of chocolates. This was a plastic box which we bought some sweets on the festival, and this is the common garden pot. So it's easy to culture them. We need to just put the soil and water them. They'll grow on themselves. And in my experiment of yeast or fermented coconut water, I will say, here we can see the water bottle, and this plastic bottle contains the fermented coconut water. And what I do is, I take this container, a plastic box, plastic small container, and I have placed a boiled potato slice here. I have placed it. So what I do for streaking them is, I take this kind of earbud kind of thing. We can even use something else like rod or like a toothpick kind of thing, a slender stick kind of thing, slender wire which is rigid kind of thing. So I just hold it in this, and I'll shake the bottle and just tilt it so the water will touch the bud, and then I will simply put in a S-shaped manner on this potato slice, as simple as inoculation, and I'll cover it with a lid to prevent it from the attack of insects and fruit flies and ants or all such things. So here the potato slice takes the role of growth medium and agar plate. The container itself becomes an incubator. Like we can say here, the bottle itself can be called as a fermenter if we take it in a conventional way. So during this experiment, I also get a lot of support and motivation and involvement of my friends and relatives too. Like my grandma or my certain friend will say, I can see similar kind of plants growing in my compound. As I said, it is a common weed. And my mother will ask, do you need a coconut today? Should I boil 3-4 potatoes for you today while making rice or dal? So it's great involvement of all the people. And I would like to show some results of my recent potato slices. So here are the results. I covered actually the potato. I was running out of the lid, so I used the empty coconut shell. So everything is possible here. So here we can see the large number of colonies which are growing in 48 hours, different colonies. And here we can see this slice is of 24 hours. So by studying the morphology and characteristics which are shown by the colony, we can get various colonies here, n number of organisms, n number of microbes grows. So by just observing it closely, the potato slices, we can compare to the colony characteristics and morphology of the bacteria or fungi, and I try to identify them. Even in case of Phyllanthus, through daily ChatShaala, I was able to identify my Phyllanthus till the species level. So some pictures will be shown now to where we can see the results. Some photos will appear now. Yes, so these are some photos. The left-hand side is the inoculation done by me. Here it is present, and the right-hand side photos shows the one done by the nuns. We can see the colonies, various colonies present over there. So it touches upon various context to the curriculum, to the syllabus, to rather I would say day-to-day life, and makes us curious about the observation, what's happening.
So in the labs also, if we see before using the microscope or any instrument to identify the colonies, we actually do colony characteristics studied in order to identify the colony or guess which colony would be present on the agar plate. So similarly, we can do it at home lab using potato as a medium. So as shown by Aditya and also through Batul Mam, we have an idea that we can grow things, we can study microbiology at home labs. As such, there would be several such sources of microbes in kitchen labs that can be studied. So coming back to fruit flies, as mentioned while discussing fruit flies were active during the day and not active during the night. Similar behavior was being observed by plants. So actually Cubists found this plant, Phyllanthus. In the picture, you can see that the plant is opening the leaf, that is referred as wake up, and the closing of the leaf is referred as sleep. So Phyllanthus is also commonly known as Bhui Amla or Bhumi Amla. It is a close relative of Avla or gooseberry plant. So the activity is closely linked with closing and opening of the leaf. And it is interesting to note that the opening and closing of leaf is not not the behavior that we observe in mimosa or touch me not plant, because mimosa or touch me not plant, when you touch the plant will close the leaf. Here during the day the leaves are open and during the night the leaves are closed. Lakshmi, can you demonstrate it for us? Lakshmi is an MSc botany student from Thrissur, Kerala.
Yes, Sayada. Hi everyone, welcome to my home lab, Mambali, Thrissur, Kerala. Here are my Phyllanthus plants which are growing in a grow bag. So I have two species of Phyllanthus plants, Phyllanthus urinaria and Phyllanthus amanus. So since Aditya said it's a weed, we can see these plants even in our roadsides, in our gardens also. So at the same time, we can also easy to culture these plants in our home lab by simply giving water to them and allow to them grow. So like as Sayada said, Phyllanthus plants are actually not like touch-me-not plant. In case of touch-me-not plant, when we touch the plants, they immediately close their leaves. So here we can see, I touch these Phyllanthus plants, but they are not closing. So they are not like touch-me-not plant. At the same time, both mimosa and Phyllanthus showing a behavior of opening their leaves during the daytime and closing their leaves during the night time. So here we can see in the daytime, all these leaves are get opened. But during night time, so in the evening time, they started to close their leaves. And in the night time, they completely go to sleep. In the next morning, they again open their leaves. So this phenomena, we call it as sleep-wake cycle. Over to you, Sayada.
So thank you, Lakshmi, for demonstrating the behavior of phyllanthus plant. So as you heard from Lakshmi that even mimosa plant shows sleep and awakeness pattern. So during the night, phyllanthus plant will also close the leaf even without touching. So here is a video shared by Susanta sir from Sappekatti, Assam. A time-lapse video beautifully capturing the opening of leaf during the day. Yeah.
Yeah, I just want to say that Sushanta is a school teacher from Assam.
Yeah, unfortunately, he couldn't be with us because he was affected by COVID recently. He's come home, he's all well, but he's on rest. So, but he has given us a beautiful time-lapse video to see the opening of the leaf. Yeah, so as you can see, yeah, and this is the closing of the phyllanthus leaf. So like I said, the opening of the leaf is during the day and the closing of the leaf is during the night. Here in the dark period, you can see the phyllanthus starting to close the leaf. So in ChatShaala, we actually tried to connect this opening and closing of leaf with the sleep-wake cycle of fruit flies. So this is the whiteboard of the ChatShaala discussion. As you can see in the graph, the red is denoting fruit flies and the green one is denoting phyllanthus. The opening of the leaf is coinciding with the increase in number of fruit fly on the tomato piece that we keep. And the closing of the leaf is coinciding with the declining number of fruit flies. So there is through collaboration, we are still continuing to understand the behavior and connect it to human sleep-wake cycle, because humans also are active during the day and sleep during the night. So yeah, through collaboration, we are still developing on it. As you can see, people from different parts of country, like there is Nainital, Muradabad, Assam, Kerala, Mumbai. Several parts are collaborating together to understand the behavior of this plant. There are several questions being asked. And one of the questions that they are addressing is light playing any role in the opening and closing of the leaf, in the sleep and awakeness pattern of this plant? Because the opening of leaf is coinciding with the sunrise time and the closing of the leaf is coinciding with the sunset time. So does light play any role is the question that Cubists are addressing. So phyllanthus is also popular for its traditional tribal medicinal value. So other than being popular for its medicinal value, phyllanthus is also quite popular for its controversies. Lakshmi, can you address the controversies?
Yes, Sayada. So actually, the researchers from India have reported their work in Phyllanthus niduri by claiming that this plant is native to India. But we found some references that suggest that Phyllanthus niduri is actually native to America. It is not found in India. So this controversy is quite popular as Phyllanthus niduri controversy. And the references also suggest that 3 different Phyllanthus species closely resemble Phyllanthus niduri. So Cubists across the country are trying to critically look into this and to find out whether the claim that Phyllanthus niduri is native to India is true or not.
So collaboratively, we are understanding whether Phyllanthus niduri is native to India or not. Although the sleep-wake pattern in Phyllanthus is studied through the phenomena of opening and closing, like opening is referred as wake up and closing of the leaf is referred as sleep. It is also very well studied in well-known plant model system, Arabidopsis thaliana. But Arabidopsis thaliana cannot be cultured at home because it needs temperature control as it is native to Europe. So in the hunt to find an alternative, so yeah, as you see the map displayed on the screen, people from different parts of the country, again, Nainital, Muradabad, Ranchi, Assam, Delhi, several other places, Cubists are collaboratively finding out an alternative. And we found an alternative cardamine, that even researchers are taking a closer look at. A weed that is found in India that is a close relative of Arabidopsis thaliana. So this photograph is showing in the left side there is Arabidopsis thaliana, on the right side it is showing cardamine. This plant is pretty similar to Arabidopsis thaliana. So this is a photograph of home lab of one of our collaborator Arunima in Kozhikode, Kerala. So this cardamine plant can be, as you can see, she is maintaining cardamine plant at her home lab. Wherever we get place, we make it a lab. So with this cardamine plant, we can understand genetic engineering. I think we all will be familiar by this genetic engineering term, at least somewhere in school you have heard about this. And a common example that is usually used in classrooms or in academic settings is BT cotton. But this process of genetic engineering that is done in BT cotton is quite sophisticated and expensive. Although in cardamine, we were collaboratively discussing that floral dip method can be used, which is rather inexpensive as compared to the sophisticated method that we were talking in case of BT cotton. So Arunima, can you tell us about it? Arunima is an MSc botany student from Kozhikode, Kerala.
Yes, Sayada. So by the term itself, the floral dip method, we can guess that the genetic engineering is done by dipping the flower bud into a solution which containing the gene of interest. And later, this flower bud will develop into a flower and then into seeds. So we expect that after dipping the floral bud in the solution which containing the gene of interest, the total seed yielded. Some of those seeds will be transformed seeds or the genetically engineered seeds. And some of the cubists like Dr. Shalini Mam and Manisha from Ranchi, Jharkhand, they were attempted this floral dip method. And the gene used here is the antibiotic resistant genes. So we expect that the transformed seeds or the genetically engineered seeds will germinate in a medium containing antibiotics, as the transformed seeds contain resistant genes, whereas the normal seeds will not germinate in a medium containing the antibiotics.
So this is when the lockdown was announced in Ranchi, and the Ranchi Cubists who attempted to do this floral dip method, they had just the transformed seeds with them. So during ChatShaala discussion, when we were discussing with the Ranchi group, we found out that Ranchi Cubists have not we have not find out the normal seeds, that are they growing in, how they are growing, are they germinating in antibiotic medium or not. So this was actually a goof up, and in Cube we celebrate goof up, we celebrate mistakes. So when we talk about our mistakes, we collaboratively discuss it and come up with solutions. So through ChatShaala discussion, one of our collaborator Nazish from Mumbai collaborated with Ranchi group. Man, a student from a graduate student from Ranchi, Jharkhand. Can you tell us about Nazish's work?
Yeah, hello again. So Najish had cardamine plants at her home lab, and we wanted to find out what actually happens to normal seeds when grown in a medium containing antibiotics. So our expectation was that the normal seeds will germinate in water, but not in a medium containing antibiotic. Surprisingly, we found that the antibiotic did not affect the germination of the seeds. Instead, it affected the color and size of the seedlings. So we see here cardamine seedlings at the leftmost was germinated in water, and the cardamine seedling at the rightmost is germinated in high concentration antibiotic. We can see the seedling is germinated, but there is a huge size difference and color difference. The seed that is germinated in antibiotic is comparably smaller in size, and the color is turned into pale green. Like so, in between these are seedlings germinated in varying concentration of antibiotics. So this was it.
So By this, we found out that the antibiotic that was used was kanamycin. So the antibiotic is not affecting the germination of the cardamine seeds, but it is affecting the size and the color of the seedlings. So cubists were also facing problems while germinating normal cardamine seeds in water. So this is when Cube group, basically nature mechanics groups from Trivandrum of mothers and grandmothers, started to address germination rate study in the seeds available at home like green gram, because generally we don't face issues while germinating these, because we use this for this sprouts for salads and stuff. So why is cardamine facing an issue? So that is when mothers and grandmothers started to critically look at the germination rate study. So along with this, cubists were also noticing cardamine plant getting eaten up by caterpillars. So there is a video from our collaborator Arunima from Kodikor, Kerala. So this caterpillar is feeding on cardamine plant. So that all plants not get affected by it, we separated this caterpillar with small cardamine plant, and so that the other cardamine plants don't get affected by it. And when this caterpillar developed into a butterfly, we identified it as Psyche butterfly. Here you can see a video Psyche butterfly on when the caterpillar actually developed into Psyche butterfly by Arunima actually. So another collaborator, Shraddha Sonavne from Panvel, Maharashtra, she was also looking at this Psyche butterfly, and she found out that Psyche butterfly feeds on Vernonia plant, that is commonly known as ironweed. So when she was finding about what is the feed for Psyche butterfly, she also found out that Psyche butterfly lays eggs on cleome plant. So when she was looking at Wikipedia pages in order to find this out, like I said, she found cleome plant is where Psyche butterfly lays eggs. But we found cardamine plant was also where Psyche butterfly was laying eggs. So clearly it shows cubists just not believe whatever is there on the internet, that Wikipedia pages needs to get updated, because even we have references, we have evidence that Psyche butterfly also lay eggs on cardamine. So we are critically looking at everything through collaboration. We just don't believe everything that is there on the internet as true. So here you can see collaborators map that are joining ChatShaala, like GN mentioned, Arunan sir mentioned, from different parts of the country and actually different parts of the world. We are growing our family. People from different parts of the world are joining the ChatShaala. Then there is a graph that shows Cubists joining ChatShaala. So this is a graph from March 2020 to March 2021, where Cubists are regularly, for 4 hours every day, they are collaboratively discussing and finding answers to questions and constructing knowledge. So we can say by this, I think we can say home lab is possible. And if it is possible in lockdown, it can be done otherwise. And like Jian also said, that every citizen has a right to develop scientific temperament. It is the right of every citizen. So like gully cricket, through frugal home labs, we are trying to understand the rules as to how to play STEM games. So let's learn from this gully cricket model and construct knowledge, learn science collaboratively, and turn our homes into home labs. So thank you. Now we can start questions and answers. We can address questions of the participants.
Thanks so much, Sayada, for coordinating this, and thank you all to all the people who shared your experiences. This has been fantastic, and the proof that it's been fantastic is the number of questions we have on both Zoom and YouTube. And so let's actually start in reverse order with some of these things which were more recent, and we will go back to the fruit fly questions a bit later. One thing is, let me just see if I can open up this chat window, and let's go to YouTube. I think YouTube, Okay, so there are lots of questions here. Of course, please remember we've put the links to join the ChatShaala thing as well. One thing about the studies on the opening and closing of Phyllanthus, a question from PU and some other people. What happens if I put artificial light? If you keep the plant under artificial light and turn the lights on and off at regular times, will the plant follow the sleep-wake cycle depending on your ambient light, or does it have something to do only with natural light?
So actually,
so I don't know who wants to answer it, but that's the question.
Yeah. So, like I said, Cubists are actually addressing this question whether light plays any role. So Lakshmi has actually tried using artificial light in order to find out whether light plays any role. Lakshmi, can you address the question?
Yes, Sayada. So actually, in our Cubists, in different parts of the country are trying to do this experiment. Not only me, Shraddha, Sunida ma'am, Chitralega ma'am are repeating this experiment by placing the Phyllanthus plants in a continuous light by providing LED light in 24 hours. So we found that these plants are actually, even though we place these plants in continuous light, in the absence of darkness also, these plants are following this pattern of opening and closing behavior. But we could find that there is a delay. The process of opening and closing is continuing, but there is a delay happens. Suppose if the plant are waking up at 6 a.m., on the next day, the plants in continuous light maybe wake up at 7 a.m. There is a delay is happening, but the process is actually continuing. And also we found that after 5 more days, by keeping these plants in continuous light.
Can I request all those who do not have, are not speaking, to mute themselves?
So, yeah. So I'll continue on what Lakshmi was saying. So we actually tried putting the plant in continuous light to find out whether this phenomena actually continues, the opening and closing of leaf, and it does. There is a delay, so light does play a role. What we have found out up until now, that light does play a role, but though it is not in the mechanism of opening of leaf. There is certain role that is played by the light, but it is not completely controlling the opening and closing of the leaf.
You're muted or no?
One more question on Phyllanthus on YouTube. I think it's Mayur who asked, since you have this collaborative network across India, do you actually see time differences across India when you, record data across India?
Yeah, so when we were talking about Phyllanthus plant, the opening and closing of leaf, the reason why I didn't mention time, instead I mentioned the sunrise and sunset time. In different parts of the country, there is time delay, there is time difference, but the pattern, the opening of leaf coincide with the sunrise time and the closing of leaf coincide with the sunset time.
Okay. There is one sort of thread which has appeared both on Zoom as well as on YouTube, and I don't know Facebook. Divya, if there are Facebook questions, just paste them into the Zoom chat so I can see them. People are a little worried about the fact that you're growing microbes, and all this, whether it is fungus, yeast, different. Are these perhaps pathogenic? What should be the safety precautions that you need? Is it okay to be growing all this stuff at home? So can somebody please comment on this?
So yes, there are some microbes that are pathogenic, no doubt about that. But like when we are using stuffs from kitchen, like using fermented coconut, we are expecting yeast to grow, because in curriculum we understand that yeast is the key factor that is used for fermentation. And other than that, even if you keep a bread slice, after some days you'll observe some colonies. So things that we are doing in kitchen, that is totally doable. Those microbes are not as effective as pathogenic. But yeah, if you try some different, like if you're trying with microbes that we work in laboratory, pathogenic, then of course we'll have to take care of it.
So basically, you're saying that if you're growing, if your bread is growing some green stuff on it, that's fine, but don't eat the green stuff.
Yeah, don't eat it. Yeah.
Okay, okay. So this actually, yeah, Jian, do you have a comment on that? You had put something in the Q&A. This actually brings us to another question, saying that I think this was, I'm just seeing if I can find the question. This was a very nice question asked by someone who said that they had left their milk. They forgotten, they drunk their milk and they forgot the glass somewhere, and after some days they found it and it had some interesting stuff growing on it, and they want to know whether it is fungus, whether it is bacteria, whether it is something. So how do you know this? And my response was that, this could be your home experiment to find out what happens when you forget your milk glass thing. But how do you figure out whether that stuff which is growing white, green, black, whatever, what are those things? Are they bacteria, yeast, fungus? Does anybody want to talk about this?
So
on potatoes and
yeah.
on whatever else?
So
Bread.
like, yeah, like we were discussing, how do we identify these microbes? What are they actually? Are they yeast? They are bacteria, or what type of yeast? How we are guessing what type of microbes, as in what types of fungus, what types of bacteria. We were discussing about colony characteristics, that this is also practiced in lab before using the microscope and stuff like that. We firstly identify or try to guess the microbes through the colony characteristics. So Aditya, can you talk about that? Because you have demonstrated that microbial study can be done at home. Aditya, yeah, can
Yes.
you give some example of how to identify this colony characteristics?
Yes. Am I audible?
Yes, Aditya.
Okay. So, like colony characteristics, as you rightly mentioned, even if before observing the microbes under the microscope, we use to guess or identify the microbe using the colony characteristic. For example, fungi, like fungus, generally have cottony, powdery growth, which spreads very much. All of majority of us must have seen the molds or such kind of fungus growing on the spoiled bread, or chapatis maybe. So generally bacterial colonies show different morphological characteristics, and also there are changes, like on the first day you may see the mold is okay, it's white in color, but on second day or third day, if it is turning black. So at least we can make a guess that maybe it is of aspergillus, maybe it is of rhizopus. And as internet is at our fingertips, knowledge is at our fingertips, so if we try to compare with Google or any papers or any material which is available on internet, so we can try to identify it. And like in bacteria, case of bacteria, majority like different microbes require a different kind of thing. Generally fungi grow in, like we say, acidic medium kind of thing. So yes, of course, fungi can be grown. And for the point of precautions, as you correctly said, no one will say the bacteria present in the curd are harmful for us, or the yeast in the wine is harmful for us. But yes, harmful microbes can also invade on our potato slices. So for that, I will as also shared by, I think, Yash in the chat. Yes, it's a right point that we are habitual now in washing hands properly after touching everything. So don't touch it directly. The potato slice, don't touch it directly by hand. Don't inhale. Smell is coming. Let's see what the smell is of. Don't put your nose near the potato slice. If we are having cut in the hand, or if we are scared of that, use our Amul bags, milk bags which comes, or polythene bags. You can put them in hand, and also keep that not in the kitchen or something. Put it in the balcony or some fixed place of the corner, so you can clean it with the soap water if you are feeling so such points can be seen. Thank you.
Although on the part of the curd, we can guess it that it would be probably bacteria, because generally in curriculum we come across that curd is how curdling takes place is because of bacteria that is present in the milk. So that through that guess we can move on to the colony characteristics. So I hope the question is answered.
Okay. Now there are okay. Let me There's one question I'm going to keep for the end because I think that's a very nice question, but let me just see if there are other questions over here. We've looked at this. Right. Dibya, are there questions on Facebook?
Mostly comments, but Arunan has already addressed those.
Okay, so let me then come to a question. There are, okay, wait a sec, there's some new chat messages. Let's just take a look at it. Okay, no, this is just comments. Let's go to Q&A. So there is a very nice question from Anushka, which I know, Gian, you've put a small comment on it, but maybe you should actually, this might be a question that everybody has. What are the basic necessities of a laboratory? What do you mean by a laboratory, right? And Gian, I think you've said something very nice, that it depends on the question, of course, but also the fact that sometimes you can do, what all can you do without equipment and just observation and things like this. So why don't you, let's end with this. What do you mean by a laboratory? What are the necessities? And how do I make a home lab,
Yeah, I mean, it's of course the most important question. I thank Anushka for asking that. See, so for example, the reason why we go to the lab is to control parameters, the variables, and that's basically one of the important things. And we want to keep on observing, taking notes, and record keeping, and then over a period of time, after collecting a lot of data, we want to study. But there are a lot of scientific experiments for which you only need your time and nothing else. For example, if you have a mango tree nearby, if you have some other tree nearby, you take pictures of the tree every week or every fortnight and actually start looking at those pictures over a period of time. I mean, you're actually doing a fantastic scientific experiment. And of course, you are not actually intervening with what's happening there. It is, in that sense, it's not an experiment. But then you will start understanding that a lot of variables are actually controlled by the climate. And now you're actually looking at and correlating those climatic conditions with what is actually happening on the plant. So what happens in August, what happens in September, what happens in November, and things like that, on that particular plant. So that requires no lab, actually speaking, and it needs just maybe a mobile camera to take pictures over a period of time. And this is one of the very popular activities that we continuously do on a regular basis in the lab. And there are many such things. Like, for example, on the other hand, if you really want to work with fruit fly, for example, as you can see, you need a lot of Bisleri bottles, mineral water bottles. And then you need rubber medium and things like that. And then, of course, you need to capture the fly and make sure that you need a corner or a tabletop or a desktop or something like that in some place in the house. I mean, that's all you need for doing that experiment. In fact, that experiment can be done at multiple places. And if you just, as somebody has shown, how to make a microscope, for example, using a glass bead and a hair clip, and now you can study those larvae at multiple stages, including starting from the egg to the entire life cycle can be studied. And of course, then the research question that you have at your hand, maybe sometimes it may need some chemicals, sometimes it may need some other things, but then a lot of these things can be done without with everything that we have at our lab. So since we are interested in science education, we actually want to say this again and again, that if you really want to learn and understand how science is done, okay, we are trying to actually say that, well, what you have at home and your neighborhood is enough to understand the scientific method. And of course, most importantly, the habit of observing and experimenting and all these things are important. But having said that, I also wanted to say one more point. I want to underline this point, which is also being said now and then during the entire presentation today. One most important reason why home labs are necessary. They are necessary because of one important impact that they do. When we are doing experiments in colleges and schools, the people at home are not involved. But then when, as you have seen, when people are using their kitchen, when people are doing these observations at home, the brother is involved, the sister is involved, the uncle is involved, the neighbors are involved, and they also keep asking a lot of questions. And in the process, they're also participating in this entire process. And this is a fascinating horizontal transfer of scientific knowledge to everybody else. I want to end with one nice story that we have. One of our school students, a Govandi girl, was actually collecting mosquitoes from the neighborhood. What she does is, she traps the mosquito and observes whether it has got white stripes on the legs or not, just to detect whether it's a dengue causing mosquito or a malaria causing mosquito. And she was doing that activity. And then one day when she returned back from the school, mother gave a small paper packet of, closed in a kind of a newspaper thing, and gave it to the daughter. And when the daughter opened, she found that the mother actually collected a lot of mosquitoes during the day for the daughter. Not only that, she even segregated those mosquitoes into dengue mosquitoes and the others. So this is the way how, this is what happens, when you actually take home labs to the home. I mean, the labs to the home. The objective of passing on the scientific temper to every citizen will happen. And this is a very important implication of what, and that's the reason why home lab, I would say, is a necessity, not just a possibility during the pandemic or something like that. It has to be done forever. Even when the lockdown is lifted, continue doing it.
Thanks so much, Jian. A quick thing, if you could clarify, there has been a question. This ChatShaala, which is, I think, more masti than any Pathshala, is this only in English? How do you take care of languages across the country? Are there local groups that are there? So we have a very large and diverse population. So how does ChatShaala happen? Somebody's just wondering about the language. Do you have to know English to be able to do this?
Well, actually, the platform can take any script of the world. So it's entirely up to you in which language you type. And, in general, what happens is, since we are people from all over the country, somebody or the other will understand your language, and they will be able to reply and will be very happy. I mean, it doesn't happen most of the time, because a lot of people actually think that, English is necessary. So please visit our site and challenge us by asking questions in your own script. And in fact, that itself will demonstrate that our platform is open for every language of the world. So the ChatShaala is, of course, open. You must have seen it already in the chat that the ChatShaala is accessible at metastudio.org, a platform maintained by Homi Bhabha Center for Science Education, for this kind of STEM games activities that you wanted to do.
Okay, on that note, it's been more than an hour and a half. Thanks so much. Thank you all for watching and being spectators today, but hopefully active participants, many of you very soon. Join in the thing. And yes, for those who've asked, why is this only biology? Don't worry. Today it is Cube. The B was biology. We will bring you more sessions covering all the sciences and mathematics. We do interesting stuff in all kinds of STEM areas. Don't worry. Today was biology. If you like other subjects, let us know. We will be glad to share what we're doing with you.
Yeah, when you actually go to ChatShaala, Arnab, when you go to ChatShaala, there are lots of chat going on in other subjects.
Right. So today you got a flavor of biology experiments. We will come back with sessions like this, telling you about all sorts of fun stuff that you can do at home, exploring the world of science everywhere, whether it's a galaxy far away or a cell inside a body. There's science all around us. Observe, record what's happening. That's how you start. Okay. And just to tell you, next Sunday, September 5th, is the first time we have a Chayanwai Sunday coinciding with Teachers' Day. So we are actually going to have a special session on that. Stay tuned and see you next time. Follow us online. join us. You can follow us on Twitter at ChaiNY, Facebook at ChaiNY. We have a YouTube channel. And if you want to get onto our mailing list, send us an email, outreach, outreach at tifr.res.in, or just Google ChaiNY TIFR, you'll find us. Thanks so much for watching. Thank you, everyone. Bye-bye for now.
Bye-bye. Thank you.